Thank you very much to the organizers for giving me the chance to talk to you today. My system is a little bit different, but I think you'll find that some of the questions that my lab is interested in have a lot of overlap with some of the questions we've just heard about. So I'm a new assistant professor at the University of British Columbia, and I work on the plant microbiome. So I think plants are absolutely incredible. They can do really amazing things, from what Kevin alluded to this morning, using their flowers to convince insects to help them reproduce. They can make leaves that photosynthesize, so they can make sugar from air and sunlight, and I'm sure we're all thinking about that since lunch is up next. They can also get nutrients which are incredibly rare in the soil. They can pump these nutrients out of the soil, and sometimes just from out of pure rock. So plants, like animals, have communities of microbes that help them perform all of these functions. So that beautiful symbiosis between flowers and insects is actually affected by microbes. So microbes can affect plant volatile production and determine which pollinators will come find them. Microbes can affect the plant immune system and photosynthesis in the leaves, and they can do this directly by the immunity function. They can do directly by competition with microbes, and they can also systemically affect the plant immune system. They can affect metabolism, both by making nutrients available in the soil that are not otherwise available, and in some cases by synthesizing their own nutrients. So here, these roots are colonized by nitrogen-fixing bacteria that can take nitrogen gas from the air and turn it to ammonia and feed it to plants. So why do we care about the plant microbiome beyond all of the cool things that it can do? It's life or death for plants. So these are soybeans that two lines that differ only in a single gene. These nice green ones can associate with those nitrogen-fixing bacteria I just mentioned. These plants have a mutation in a single gene that renders them unable to associate with these bacteria. These plants will go on to reproduce and feed the people that are growing them, and these will not. In the developed world, we can actually overcome genetic deficiencies like this with fertilizer. So we can just add nitrogen fertilizer and eliminate the need for this. However, fertilizer is really the largest ecological and economical burden of modern agriculture. And plant-associated microbes can at least help, even if they can't eliminate the need for these chemicals. In my lab, I focus on the plant root-associated microbiome. So these are the microbes that associate with the underground root parts. We call it the rhizosphere microbiome. Rhizo means root. And I think this is also a model for host-associated microbial communities. There's a quote I'm rather fond of from Daniel Jensen, that plants wear their guts on the outside. So plant roots are functionally analogous to the animal gut. It's the site of nutrient uptake, and they're exposed to the microbes in the soil, just as our gut is exposed to the microbes to the food that we eat. Plant roots, because they're on the outside, we can actually watch in real time as microbes colonize, making this a really appealing system. So plants form consistent communities of microbes. So if you take the same genotype of plant and you put it in the same soil, you get the same community back. They're complex, so about an order of magnitude more complex than the animal gut. And like the animal gut, the largest effect seems to be the environment, so the diet of the animal or the starting microbes in the soil in the case of plants. But there's a small but consistent genetic effect. So if you take two closely related plants and put them in the same soil, you see consistent genetic changes. And plants are naturally colonized by model bacteria. So this is an aerobic environment. And bacteria like Pseudomonas, Bacillus, and Streptomyces, which are a lot of our workhorse lab bacterial strains, are naturally associated with plant roots. So today I'm going to tell you about the work of my lab, studying mechanisms and host microbiome interactions. The main questions that I'm interested in are how does the host genotype affect this community structure? And then how does the structure, the microbes that end up there, affect host health? So these are very similar to the questions that a lot of people are asking in animal and other systems. For my work, I use Arabidopsis as a model. I'm sure many of you are familiar with it. It's our reference plant. So there's a lot of tools available for forward and reverse genetics. We can order mutants. and do genetic screens. It's our validated reference plant, so it's similar to other flowering plants. What we know about Arabidopsis is largely translatable to agriculturally and ecologically important plant systems. And for my work, a huge asset of Arabidopsis is that we have collections of naturally occurring accessions. So these are naturally occurring lines of Arabidopsis that occur across the globe. And we've taken 1,001 of these and sequenced them. So we have genomes of 1,001 in these naturally inbred lines of Arabidopsis. And each of these has co-evolved in its local environment with its microbiome. So we can probe the natural diversity of plant genotype and how it associates with the microbiome that would allow it to adapt to a given environment. So for the work I'm going to tell you about today, I'm mostly focused on a monoassociation with Arabidopsis and Pseudomonas fluorescens as a way to probe plant genotype. And then, based on what I find with this single plant microbe association, zoom back out to the complex community and see if what we find with this one-on-one association holds true. So Pseudomonas fluorescens, I'm sure many of you are familiar with it. It gets its name because it makes this fluorescent pigment under iron-limiting conditions. Yeah, it's easy to pick out when you plate rhizosphere samples. It's enriched in the plant rhizosphere, so it's a pretty small component of the rhizosphere microbiome. It's about 1%, but it's consistently enriched over bulk soil, so this outer circle are microbes in the soil. It's known to be beneficial to plants. It performs a variety of functions, including nutrient uptake and protecting plants from pathogens. And it's tractable, so all the tools that are available to work with Pseudomonas aeruginosa and other Pseudomonas species can be used with Pseudomonas fluorescens. So to probe the interaction of Arabidopsis with Pseudomonas fluorescens, I developed a high-throughput screening system where I can grow Arabidopsis in 48-well plates. So these are 2-week-old seedlings that are grown in liquid media with their roots submerged in the media and their leaves above, and they're separated by a little mesh disc. I can then add Pseudomonas fluorescens expressing GFP or other fluorescent bacteria into the wells, let them colonize the plant, and then read fluorescence with a plate reader as a proxy for bacterial growth in the rhizosphere. Using this assay, I screened 196 of those Arabidopsis successions that I just told you about, and I found that, in fact, there is natural variation in Arabidopsis' ability to associate with Pseudomonas fluorescens. So here is log CFU per well. And these are the accessions plotted on the x-axis, and you can see there's about a log difference in the ability of Arabidopsis to associate with Pseudomonas. Col0 is our sort of wild-type reference accession, although these are all wild-type naturally occurring strains. And I'm going to, for the next few slides, focus on these 2 Arabidopsis accessions, NOX18 and RS10, that support less growth of Pseudomonas than Col0. So I found that they support less growth in this very reduced hydroponic system. Is this true of a complex, when there's a complex microbial community present? So to ask that, I planted these 3 Arabidopsis genotypes in natural soils and looked at both their culturable microbiome and microbiome by 16S sequencing. So what I'm showing you here is the culturable microbiome from Colombia, plated on iron-limiting conditions, and then from NOX18 and RS10. And you can see that there's an enrichment in fluorescent colonies in Colombia. So there is more Pseudomonas fluorescens on Colombia in natural soil. When we sequenced the bacterial microbiome by 16S, we sort of surprisingly found that the differences were largely limited to the family Pseudomonidaceae. So between Colombia and NOX18 RS10, the majority of the community structure is intact. There isn't this large or higher order taxonomic shift like in all proteobacteria. It's pretty limited to the Pseudomonidaceae, and that was somewhat surprising. So is this very small change in community structure sufficient to actually cause differences in plant health? And so we have a number of assays that we can use to look at that. I'm just going to show you one example. But in fact, it is. So this is an assay looking at plant survival in response to challenge by a fungal pathogen. So here we're adding increasing fungal cells, and this is plant survival. So Colombia is the solid black line here, just with low levels of Pseudomonas fluorescens. And then if you add any one of a number of Pseudomonas fluorescens strains, you get protection from this pathogen. NOX18 and RS10 that don't support Pseudomonas fluorescens growth are not helped when it's present. So it's the complex interplay of plant genotype and microbe that ultimately determines plant health. So because Arabidopsis is tractable, we can actually map these changes. So NOX18, RS10 that I've been telling you about, there are single loci that are responsible for the majority of the difference. I'm not quite ready to tell you what the genes are, although we have some good candidates. But I can tell you that these NOX18, RS10 seem to be making something that looks like an antimicrobial. So these are bacteria growing just in the exudate, so just in the what's secreted by the plant root on the Columbia plants. And so Pseudomonas fluorescens stained with DAPI, you get these sort of normal-looking little bacteria, and then they're counterstained with Cytox, a membrane-impermeable dye. So most of the cells are alive with impermeable membranes. In the NOX18 and RS10 rhizosphere, you see this dramatic change. You see filamentous bacterial cells. Most of them have permeabilized membranes. And in some cases, you can see these membrane blebs, which you probably can't see too well here. But those are often characteristic of antibiotics that disrupt bacterial cell walls. And so what it looks like we have here is an antimicrobial that is specific to Pseudomonas, right? That's the only shift in the rhizosphere microbiome. It's just Pseudomonaceae. So going forward, we're using a combination of genetics to map the genes and then biochemistry to try to identify this putative novel antimicrobial. So for the second half of my talk, I'm going to focus more about how microbiome composition affects host health. So I started getting into this because closely related Pseudomonas strains can have really dramatically different effects on plant health. And I'll give you an example to illustrate this. And this is an assay called where you can look at systemic defenses against a pathogen. So here we treat plant roots with Pseudomonas fluorescens and then challenge leaves with a plant pathogen. In this case, I'm using Pseudomonas syringae, so it's a little confusing because it's two different Pseudomonas. But this is a plant pathogen, Pseudomonas syringae, and the fluorescens are the beneficial microbes. So we found that fluorescens in these assays can either induce resistance to this pathogen or susceptibility, and that's dependent on which strain is present. So here's an example of what these data look like. On the y-axis is growth of Pseudomonas syringae in the leaves, number of cells per centimeter squared of leaf. Our plants, if you treat them with buffer, you get about 10 to the 7th cells per centimeter squared. If you treat with this strain that induces systemic resistance, or ISR, you get a reduction in bacterial load in the leaf, indicating these are more resistant. If you treat with these ISS strains, you get an enhanced bacterial load in the leaf, indicating they're more susceptible. And if you actually just look at the leaves, it's pretty obvious. So buffer-treated plants, you get some disease symptoms, this chlorosis. If you treat with these ISR strains, you can see the leaves look greener, they look better, and ISS strains, you see significantly more disease. So the really interesting thing here is that these strains are indistinguishable by 16S. So they're more than 97% identical. So this is sort of a perfect opportunity to figure out, so basically you can't predict microbiome function by 16S, right? If you sequence 16S, you can't tell which strain is there. You can't tell what the health outcome is going to be for the plant. And here's a phylogeny of a number of these strains that we've tested and now have full genome sequences for. The ISS and ISR strains within Pseudomonas fluorescens and a close sister clade, Brassica cearum, are boxed in orange and blue, and you can see that they're dispersed throughout this clade. So again, phylogeny is not predictive of function. So I've gotten really interested in figuring out what these bacteria are doing to the plant, how is it that they're changing plant defense responses, and how is the plant perceiving these different bacterial strains that result in such different phenotypes. So I'd like to say that we use some really fancy high throughput assay or fancy equipment to figure this out. And this was actually, in reality, it was just a case where I stared at a lot of plants for a lot of years and started noticing that there were differences in the way the plants looked, depending on what we treated them with. So if we treat them with buffer, and these are sort of extreme examples just to illustrate the point, the plants are a little bit smaller. If we treat them with ISR bacteria, they're bigger, but not quite in the same way as with these ISS bacteria. So the ISS bacteria that are making plants more susceptible, the plants are bigger, they're greener, they have this really bright green color, the leaves are flatter. These ISR strains, it's hard to see here, but they actually make the leaves a little darker. They look a little more like leafy green kale. And Arabidopsis is related to kale. So I started thinking maybe they're changing, not just making the plants bigger, but changing the nutrient composition. And so I sent the plant tissue off to a number of like public state school, ag schools. For farmers, will test nutrient composition of their plant tissue to look for nutrient deficiencies, and they were kind enough to test my Arabidopsis tissue. They tested it for a sort of standard panel of nutrients, and the only difference we found with their assays was in nitrogen. So the ISS strains seem to enhance slightly but significantly the nitrogen content of the plant shoot relative to buffer-treated plants, and the ISR strains actually seem to deplete nitrogen. There's less nitrogen per gram of tissue. So for the ISR strains, we actually didn't find any differences using these assays, but they actually mostly look at macronutrients. And this sort of dark leafy green color made me think of iron, because that's, our kale and our leafy green vegetables are really high in iron. So I turned to a lab that focuses on iron and other micronutrients in plants, which is Mary Lou Garneau's lab at Dartmouth. And I've been working with her graduate student who got this assay working in plates where she can grow plants under iron-limiting conditions and then adds the ISR bacteria, and you can see rescue of iron deficiency symptoms. So the plants are sort of chlorotic, under low iron, and the bacteria rescue this. And she used ICP-MS and found that, in fact, there is an enhancement of iron content of the plant tissue when you treat with these ISS bacteria. So by going through a large number of strains, we started to see these patterns emerge where you have strains that promote resistance or susceptibility, and that correlates with the growth promotion phenotypes, where some of the strains promote iron uptake and some promote nitrogen uptake. And so that led to the hypothesis that maybe the bacteria are affecting plant disease resistance by affecting plant metabolic state. So by affecting plant nutrient uptake, by affecting nitrogen uptake, they're making plants more susceptible. By increasing iron uptake, they're increasing resistance. So we tested whether changing plant nutrient status was sufficient to phenocopy these effects, and that's shown here. So if you add the ISS bacteria or nitrate, you can see that plants become more susceptible to a pathogen, Pseudomonas syringae. And similarly, if we add the ISR strain or iron, plants become more resistant. So at least changing nutrient status is sufficient to phenocopy this. Again, supporting this idea that maybe it's through changing plant nutrient status, we're changing plant immune responses. So why might changing nitrogen or iron status affect plant disease resistance? First, I'm going to tell you about the iron response, because we have a slightly more compelling story for that. It's known from macrophages and other, you've probably heard of this from macrophage work, that iron can catalyze the formation of reactive oxygen species through Fenton chemistry. So iron and hydrogen peroxide can be converted to reactive oxygen, free radicals. And it's known that reactive oxygen production is the first line of defense in plants. So when they perceive bacterial flagellin or LPS, there's an oxidative burst. But iron had not been previously implicated in this process. So we tested whether iron could induce reactive oxygen production in plants. So this is an assay where we use a luminol reporter. So it's a luminescent substrate that becomes luminescent in the presence of an oxidizing agent. If you add bacterial flagellin, so FLAG22 is a peptide that induces defense responses in plants. FLAG22 will elicit an oxidative burst in plants. If you just add iron, you don't get any response. But when you combine the elicitor and iron in a dose-dependent manner, you get increasing production of reactive oxygen, indicating that iron can participate in Fenton chemistry in enhancing ROS production in plants. Or maybe not in Fenton chemistry, but it can participate in ROS production. With the Garneau lab at Dartmouth, we tested a number of iron uptake mutants. So some of these mutants overaccumulate iron, and some of these plant mutants have less iron. And we found that there's a correlation between plant iron content and oxidative burst. So OPT3 and ISR2 have higher iron content than wild type here. And then IRT1 is actually the plant iron transporter, and so this has less of an oxidative burst. So collectively, this suggests that it's by inducing plant iron uptake that bacteria in the rhizosphere are enhancing plant pathogen resistance. So for the nitrogen story, we actually don't have as compelling a story as my best guess is that plants with higher nitrogen content are just tastier to bacteria, but it's also possible that there's something more complex signaling related going on. So now I've actually started playing with what happens if I mix these strains together and then change plant nutrient content. Does that change the community structure and the community function? So first, I actually found that as much as I wanted it to, I couldn't get nutrient treatment to change bacterial abundance in the rhizosphere. So here I'm mixing both the ISR and the ISS strains, and then changing nutrient abundance, and then we just plate the bacteria because we can't tell these apart by sequencing. So strep resistance or RIF resistance in the different strains, and there really aren't any significant changes under different nutrient conditions. However, we do see changes in plant defense response dependent on the nutrient status of the plant when both strains are present. So if you add iron, the plants are more resistant. When both strains are present, when you add nitrogen or iron and nitrogen, the plants are more susceptible. And so we can start playing with these complex interplay of plant genotype, bacterial genotype, and the environment. So far I've told you that Arabidopsis accessions can inhibit the growth of Pseudomonas in the rhizosphere, and it looks like by producing a Pseudomonas-specific antimicrobial. And Pseudomonas affects plant immunity by altering plant nutrient status. So what my lab is currently actively working on is trying to find what the functional unit in the microbiome is. So what bacterial genes are actually required for altering plant nitrogen or iron status. And so for that, we're coupling functional assays with comparative genomics, so whole genome sequences of these strains, and then comparative genomics to identify genes that are unique to the ISS or the ISR strains. And using this approach, we've actually found a fairly small number of genes that are unique to the ISS strains, so 25 genes and 10 operons, and ISR is a little bit larger. Right now, we don't have a sense of whether ISS is due to loss of function of some of these genes or gain of function of these genes. So we're coupling that with transcriptional profiling and some functional assays. So I'm going to show you one last data slide, because this is the very first data to come out of my own new lab, which is very exciting. So my student, Paulina, has been starting to just look at differences in nitrogen metabolism in these different strains. So she's just growing them. These are just growth curves with different nitrogen sources for either this is the ISR strain or the ISS strain. The ISR strain can grow, they actually all grow very well with glutamine as their sole nitrogen source. The ISR strain can grow with ammonia, and it actually can do this amazing thing that some Pseudomonas fluorescent strains can do, which is to use nitrate as a sole nitrogen source under aerobic conditions. So it can reduce nitrate even under aerobic conditions. These ISS strains, and actually she's tested a lot of them now, they use ammonia very poorly and nitrate even worse or not at all. So it seems like there are some differences in nitrogen metabolism that at least correlate with the phenotypes we're seeing on the plant. So it might be something as simple as this strain is sopping up all of the available nitrogen. At least these are the preferred form for the plants, whereas these are leaving it available. The other finding that my students made, which is this was the very first thing that I would call new data from the lab. So all of these strains were identified because they make this fluorescent pigment on plates. They make pyroverdin, this iron chelator. I had never actually shown a black light on the cultures, but the students, because these bacteria fluoresce, they shine a black light on everything. And so we found that these ISR strains make the iron chelator under lower ODs in culture. So they turn this on way before these ISS strains do. So it seems like, and this is specific to culture, they all seem to turn on the genes at the same time when you just streak them on a plate. So it seems like there's some differences in regulation and production of this iron chelator that also seem to correlate with the iron and nitrate uptake phenotypes. So with that, I'd like to thank my small new lab, my 3 students, UBC and Michael Smith Labs in the Department of Microbiology to help me get off to a great start there. My postdoc advisor, since I'm a new fledgling, is still giving me a lot of support. And then I have 3 wonderful collaborators, so Lori Shapiro in Naomi Pierce's lab, who's been doing the comparative genomics, Amanda Socha and Mary Lou Garano that have been helping with the iron stress and iron uptake, and then Buck Samuel from Baylor College of Medicine that helps with the 16S microbiome sequencing. Thank you.